Welcome to the Bioprocess Pulse. This is not a news recap. It's an intelligence brief. Each week, we strip away hype and novelty to focus on what actually moves cost curves, survives scale-up, and can be built into real fermentation and biomanufacturing assets. If you're a scientist, founder, or manufacturing leader who cares less about headlines and more about deployability, you're in the right place. Let's get into this week's signals.
Welcome to The Debate. I'm your host. Today, we are dissecting the Bioprocess Pulse Intelligence Bulletin for the week of February 5th through the 12th, 2026. And the theme this week is stark. We are done with moonshots. We are not talking about platforms that might work in 2030. We are looking at deployable signals, processes, and hardware ready to bend cost curves right now.
And that is exactly where I'm going to, stop you. Deployable is a dangerous word in this industry. I've analyzed the same bulletin. Where you see optimism, new sensors, process tweaks, regulatory shifts, I see a landscape just littered with traps. You see signals that reduce costs. I see signals that expose just how brutal the physics of scaling really are. Moving from a petri dish to stainless steel is rarely a deployable plug-and-play event.
The skepticism is fair, but look at the macro data. This week suggests we are finally exiting the era of genetic heroics, where we tried to solve everything by rewriting the genome, and entering an era of process discipline. The industry is proving that we can collapse constraints like toxicity and cell density using standard equipment. These are immediate economic wins.
You call it process discipline. I call it the physics of the tank. The bulletin highlights impressive lab results, sure, but when you scale those deployable wins up to 100,000 liters, you hit hard limits, aeration costs, mixing heterogeneity, and frankly, massive data burdens. The transition to commercial reality is where these signals usually die. But let's start at the top. You think the market is stabilizing.
I do. Look at the headline acquisition. Eli Lilly buying Orna Therapeutics for $2.4 billion? That isn't just a purchase, it's a statement. Orna does in vivo CAR T-cell therapies. Lilly isn't buying a bespoke, patient-specific autologous therapy that requires just a logistical nightmare to manufacture. They are buying an off-the-shelf solution.
It's a move away from the clinic and toward the factory.
Exactly. They are trading clinical complexity for upstream complexity. They are betting that if you can engineer the viral vector scale-up, you win the market because the delivery is simple. It's a massive vote of confidence in manufacturing realism over personalized medicine.
See, I view that $2.4 billion differently. It's not confidence, it's pressure. Big Pharma is terrified of their current cost structures. Look at the other major signal. Sanofi replacing CEO Paul Hudson with Belian Garjo from Merck KGaA. You don't swap out your CEO if your R&D engine is purring. Garjo is an operations veteran. She understands the nuts and bolts of efficiency. This tells me that Sanofi knows their R&D productivity is lagging, and they are bringing in a fixer to tighten the screws on the bottom line.
I wouldn't call it panic. I'd call it a strategic pivot to what works. Look at Bridge Bio. Their phase 3 success with infigratinib for achondroplasia, that's an oral small molecule beating out complex injectables. The market is rewarding simpler delivery and simpler manufacturing. The winners aren't the ones with the flashiest mechanism of action anymore. They are the ones who can produce a pill or an off-the-shelf therapy at scale. Manufacturing economics are becoming the decisive factor in market share.
Economics are decisive. I agree. But that's why I'm worried. You look at a company like Kane Biotech restarting commercial activity in North America, and you see momentum. I see a company walking back into the fire. The technical bar for execution has risen. It's not enough to just have a product. You have to survive the margin pressure. And that brings us to the core of the debate today. The upstream inflection. You seem convinced that we've, broken the biological constraints in the fermenter.
We have. The evidence is right there in the E. coli data for GLP-1 production. For the audience, GLP-1 is the class of drugs used for weight loss and diabetes. Huge demand. This week's data is the strongest evidence I've seen that process engineering beats genetic over-engineering.
Walk me through it, because I saw the numbers and they look suspiciously high.
They hit nearly 90 grams per liter of dry cell weight and about 10 grams per liter of soluble fusion protein. For a recombinant protein, those are massive numbers. And the kicker? They didn't use AI to redesign the genome. They simply controlled the process.
Process control is broad. What was the specific lever?
Temperature and timing. They delayed glucose feeding by 4 hours to let the biomass develop respiratory capacity, basically getting the cell's lungs ready. But crucially, they dropped the induction temperature from the standard 37 degrees Celsius down to 25 degrees.
A 12-degree drop.
Correct. By dropping the temp, they slowed down the protein folding. This suppressed the formation of acetate, a common waste product that slows growth, and prevented the protein from clumping into useless aggregates. They took a standard host, applied rigorous kinetic controls, and achieved a result that drives a 20 to 30% cost reduction. In a market where GLP-1 prices are crashing toward what? $149 a month? You survive by optimizing these wrong variables like temperature.
That's a compelling argument for the pilot plant. But have you considered the thermodynamics of that tank? You're talking about an OD of 180. Do you know what the oxygen demand, the OOR of E. coli is at that density? It's voracious. You can drop the temperature to 25 degrees, which helps with the cooling load, sure. But the viscosity at 90 grams per liter is like mixing wet cement.
But the productivity gains offset the mixing costs.
Do they? In a 100 cubic meter tank, you are going to have massive oxygen gradients. You physically cannot push enough air into that cement to keep every cell happy. You will have pockets of starvation. And let's look at the other story in the intelligence briefing, the 5-ALA, the toxic intermediate story. This is where your process discipline argument starts to crack.
The 5-ALA case supports my point. They push titers to 37.3 grams per liter for a molecule that is usually toxic to the cell. They use systems, metabolic engineering, and quorum sensing to delay production until the biomass was ready.
Quorum sensing. That is the phrase that keeps me up at night. For those listening, quorum sensing is a biological switch where cells wait until they reach a certain population density before turning on a specific gene. It works beautifully in a 5-liter glass reactor. The signal molecule hits a threshold and bam, the whole population switches together.
It's an elegant solution to toxicity.
It's elegant in the lab. In a 100,000-liter tank, it's a gamble. Mixing times in those giants are measured in minutes, not seconds. You get hot zones where the signaling molecule is concentrated and dead zones where it isn't. You get what we call the popcorn effect. Half your population switches to production mode while the other half is still growing. You lose that synchronization. You haven't collapsed constraints. You've just introduced a new one, the hydrodynamics of the vessel.
You are assuming we are flying blind in those large tanks. You're assuming we can't see those gradients. But the hardware signals this week prove otherwise. We aren't just relying on a single pH probe on the wall anymore.
You mean the fiber optic sensing?
Exactly. The deployment of distributed fiber optic oxygen sensing is a game changer. We have commercial data now from fermenters larger than 100 cubic meters, and the finding was critical. The failure mode wasn't low average oxygen. The failure was oxygen fluctuation.
Explain that.
As the cells circulate through the tank, they pass near the impeller, high oxygen, and then drift to the stagnant corners, low oxygen. They cycle between hypoxia and hyperoxia. This cycling stresses the metabolism.
Which proves my point about the gradients.
No, it proves we can now manage them. With distributed sensing, coupled with model predictive aeration control, we can see the dead zones in real time. We can modulate agitation to smooth out those fluctuations. It converts scale-up from a black box gamble into a control problem. This acts as a force multiplier for the process.
Sensors are great, but I don't buy that they fix the underlying biology. It's a classic engineer's fallacy. If I can measure it, I can fix it. Let's look at the continuous biocatalysis stress test mentioned in the briefing. This was an enzymatic process. In the lab, the enzyme had a half-life of 500 hours. It was rock solid.
In a clean buffer solution, yes.
Right, but when they moved it to a pilot-scale, real-world conditions, it failed. Why? Because of trace aldehydes coming from the upstream fermentation and fouling at the membrane loops. The intrinsic stability of the enzyme didn't matter. The impurities in the real-world broth killed it. A fiber optic cable telling you the oxygen is fluctuating doesn't remove aldehydes.
But they solved it. They used a scavenger resin to pull out the impurities.
They did, but look at the cost. They had to add a whole new unit operation, a scavenger resin column, and they had to reduce the enzyme loading. That destroys your unit economics. My point is that deployable implies readiness, but the stainless steel environment introduces chemical aggressors, impurities, shear stress, fouling that your lab models completely miss. You end up patching the process with expensive fixes like resins.
That's a fair point on the enzymes, but you're ignoring the regulatory tailwinds helping us absorb these costs. The governance landscape is shifting to subsidize these risks.
Are you talking about the FDA and EMA guidance?
Yes, the EMA's work plan for 2026 and the FDA's alignment on ICHQ-13. For the listeners, ICHQ-13 is the global guideline for continuous manufacturing. Regulators are treating continuous processing as a reference standard now, not a risky outlier. This de-risks those hybrid architectures you're worried about.
They are rolling out the red carpet, but there is a trapdoor underneath it.
How so?
Have you read the fine print on ICHQ-13? It demands intense digital data integrity. You cannot run a continuous line with paper batch records. You need real-time traceability, sophisticated process analytical technology, PAT, and a data infrastructure that can withstand a forensic audit.
That's modernization. It's necessary.
It's necessary, but it's expensive. It's great for Sanofi or Lilly, but for a startup, for a small biotech trying to be deployable, it's a massive barrier to entry. The regulatory bar for data infrastructure has effectively gone up. You can't just hack together a continuous process anymore. If you don't have the digital backbone, the FDA won't touch you.
I disagree that it blocks smaller players. Innovation finds a way. Look at MicroHarvest. They are a great example from this week's intelligence. They didn't just try to build a factory in a field. They utilized a German federal grant for energy efficiency and co-located at the Luna Industrial Park.
MicroHarvest is a fascinating case study, but it proves my point about physics, not policy.
They solved the CapEx problem by sharing utilities. That's smart business.
They had to. Look at the postmortem on their scale-up. The original assumption was cheap feedstock. They thought growing biomass on waste streams would provide a margin cushion. But what happened at 15 kilotons per annum? The aeration and cooling costs dominated the OpEx.
Energy costs are always a factor in fermentation.
It wasn't just a factor, it was the whole story. The cheap feedstock advantage was completely wiped out by the electricity bill for the compressors. The mass balance showed that oxygen demand drives the cost. The MicroHarvest example proves that unless you solve the heat removal and gas transfer at 15,000 tons, your cheap carbon source is irrelevant.
That's exactly why the co-location strategy worked. They adapted. I see MicroHarvest as a success story of policy support. The government grant effectively subsidized their process limitations to get them across the valley of death. We are seeing a trend where policy tailwinds are helping companies survive the physics.
Policy can help, but it can't change thermodynamics. If you are relying on a grant to pay for your electricity because your organism needs too much oxygen, you don't have a sustainable business. You have a subsidized experiment.
That's too cynical. The combination of these signals, the process tweaks like the GLP-1 temperature drop, the advanced control from fiber optics, and the regulatory acceptance of continuous manufacturing creates a toolkit. A founder today has more levers to pull than they did 5 years ago. The distance between insight and execution has shortened.
The distance may have shortened, but the road has gotten rougher.
So let's distill this. I'm a founder or an engineer reading this bulletin. What is the takeaway? I say the takeaway is action. The 20 to 30% cost reductions are there. You don't need a new strain. You need to implement delayed feeding. You need to drop the temperature. You need to install the sensors. The signals are deployable now.
My takeaway is caution. Deployable requires resilience, not just optimization. If you optimize your process to the razor's edge like that OD180's GLP-1 process, you had better be sure your tank homogeneity is perfect. If you rely on quorum sensing, you better have a plan for signal desynchronization. The winners won't be the ones with the best lab data. They will be the ones who respect the robustness required for stainless steel.
But you have to admit, the industry is moving away from flashy biology. We aren't talking about obscure metabolic pathways anymore. We are talking about heat exchangers and data integrity.
On that we agree. Flashy biology is out. Operational realism is in. But realism hurts. Realism means acknowledging that impurities kill enzymes and that continuous manufacturing requires a digital army.
And I believe the industry is ready to pay that price for the efficiency it brings. We are seeing the convergence of biology and hard engineering.
Let's hope the engineering keeps up with the biology, because the tank always wins in the end.
That's all the time we have for this week. It's clear that whether you view these signals as opportunities or warnings, the status quo in bioprocessing is shifting.
Indeed. Read the bulletin, check your mass balances, and don't trust a sensor until you've tested it in broth.
Thank you for listening to the debate. We'll see you next week.
This week's takeaway is simple. The advantage is shifting to teams who design biology, hardware, and governance together. The breakthroughs that matter now aren't louder. They're more controllable, more predictable, and easier to finance at scale. If something you heard today challenged an assumption you've been carrying into the lab or the boardroom, that's the point. Stay rigorous. Build what scales. You've been listening to the Bioprocess Pulse. Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.